Thank you. Thank you very much, Camila. Thank you for the introduction, and thank you for the invitation to present in this forum. And thank you to everyone who's attending. My presentation will be about an application of the targeted emulation framework to meet a regulatory requirement by the European Medicines Agency. This talk will have two parts. In the first part, I will reflect on a couple of documents published by the EMA and by the FDA related to the use of real-world data. And this first part will be brief. And on the second part, I will develop the application of this study, how we design it and which problems we encounter when we implemented it and how we try to solve those. So in this first part of the talk, I will reflect on the views on causal inference for real-world evidence drafted in guidelines by the EMA and by the US FDA. As you might be aware, in 2024 and 2025, we had the publication of these two documents. The EMA published in April 2025 a reflection paper on the use of real-world data in non-interventional studies to generate real-world evidence. And in March 2024, the FDA published a document about considerations regarding non-interventional studies for drug and biological products. In the EMA draft guideline, it's interesting to see that the guideline classifies non-interventional studies into two categories: studies with descriptive objectives and studies with causal objectives. What's interesting here is that, in my opinion, they are missing a third type of studies, which are those studies with predictive objectives, but that's okay. But what's really useful here is that they start using the causal language upfront. They state that for non-interventional studies with causal objectives, the target trial emulation framework should be considered as a strategy that uses existing tools and methods to formalize the design and analysis of this type of studies. And they continue elaborating why they consider the target trial emulation framework useful. And we know this. It's because it helps the investigator consider potential biases that are related to the study design. It provides a high level of transparency on the study design, and also highlights that the alignment of time zero eligibility and treatment assignment avoids some type of biases like prevalent user bias and immortal time bias. And also remarks that despite using this framework, you still need to control for confounding. Of course, the target emulation framework was never about solving problems that derive from the data themselves. Target emulation doesn't solve measurement error, doesn't solve measure confounding. It solves problems that derive from study design. Regarding the FDA draft guideline, they state that real-world evidence can support regulatory decisions, which is an important point, if appropriate causal inference is supported by reliable and relevant real-world data. Of course, you need not only appropriate methods, you need appropriate data. And this draft guideline lists several study attributes that should be provided by the sponsor to enable the FDA evaluating the proposal for this type of studies. And among these study attributes is the proposed approach to support causal inference, for example, target trial emulation or other conceptual approaches, and to address confounding and other type of bias. So they mention target trial framework as one of the approaches that you can use to support causal inference. But they also make it clear that they don't support a specific framework. You can use any one you want, and target trial emulation framework is just one of those. So why am I starting this talk with these two documents, and why is this important? Because these two regulatory agencies explicitly acknowledge the causal goal of some non-interventional studies using real-world data. And being explicit about the type of data science tasks that we are implementing in our study will reduce the ambiguity and the scientific question, will reduce errors in the data analysis, and will reduce excesses in the interpretation of the data. And we saw that the target trial emulation framework is recommended, the EMA, and is referred to by the FDA. So with this context, let's jump into this case study. This case study is about a drug for prostate cancer. And I'm going to give you some context about this type of tumor because we will need some clinical context to interpret the results and the problems that we found. Prostate cancer is a tumor that originates in the prostate and has two characteristics. First, it's very hormone dependent. So most of the treatments and the first line of treatment consists on hormonal blockade. Hormonal blockade is different from chemotherapy, which is another type of treatment for this drug, in the sense that it's much less toxic than chemotherapy. And another characteristic of this tumor is that it has an appetite for bone metastasis. The main site of metastasis is the bone, which, as you can imagine, compromises quality of life a lot because it's painful, it causes pathological fractures, and it can affect the nerves that go close to the bone. So it's a serious clinical complication. The study that we are going to review is about a drug called radium-223, which is a drug that was designed specifically to treat bone metastasis, not to treat the tumor overall systemically, but only the bone metastasis. Why is that? Because this compound has a high affinity to go to the bone and is radioactive. So when patient receives this compound, is distributed to the bone, and that is where it exerts its therapeutic effect. This drug was approved for treatment of castration-resistant prostate cancer based on a trial, a pivotal trial, phase 3 trial called ALSIMCA, where this drug was compared to placebo in patients with prostate cancer who had received chemotherapy. That means that they were in advanced stages of their disease. They already received the usual treatments that are given as first-line treatment, so they were in later lines of treatment of the disease. In this pivotal trial, the results were that radium-223 compared to placebo improved survival and lengthened the time to first symptomatic skeletal event. So the time free of bone complications was longer than among those who received the placebo. So, and that was the basis for the approval of the drug. The next logical step for this drug was trying to see whether administering this drug earlier in the natural course of the disease was still beneficial for patients. In other words, instead of waiting for patients to receive several lines of treatment and then giving them this drug to treat bone metastasis, we're advancing it in the line of treatment could also offer some benefit. That's why the second trial was designed, the ERA-223. And in this study, they compare the combination of abiraterone, which is a standard first-line drug for prostate cancer, combination of abiraterone with radium-223 versus abiraterone alone. And they need to unblind this trial. They did that in November 2017 because they observed an imbalance in the number of fractures. In both groups, there were more fractures in the combination arm therapy, and also more deaths in the group receiving the combination of radium-223 and abiraterone. As a consequence of that, the trial was unblinded, and the PRAC from the EMA changed the indication and imposed some conditions for the marketing authorization of this drug. One of the conditions for the marketing authorization of this drug was implementing the post-authorization safety study that we are going to discuss. This post-authorization safety study was called PRECISE, and the objective of this study was to estimate what was the risk of fractures and death of patients who had been receiving this drug, Xofigo, for the time that this drug had been in the market, and whether the risk of fractures and the risk of death was different from people receiving other standard of care. We wrote this protocol in 2018, 2019, and it was endorsed by the Prague in November 2019. As every study for the EMA, it was uploaded to the EMA catalog, and there you can, with the number that you can see here, you can access the protocol, the report, and we also have a publication that is open source, and you can read it. So everything is publicly available. Everything that I'm going to discuss is publicly available. So the objectives of this study were pretty straightforward. The primary objective was to estimate the effect of radium-223 on the incidence of bone fractures compared with other standard of care for patients with metastatic castration-resistant prostate cancer. And the secondary objectives were to estimate the effect of radium-223 on overall survival and on prostate cancer specific survival compared with the standard of care. So the first step to design the study was, of course, filling in the target cell emulation table. For the target cell specification, we started with the eligibility criteria, and patients for this study need to have histologically confirmed adenocarcinoma of the prostate. The tumor had to be castration resistant, which means that they have progressed to first-line androgen deprivation therapy. And patients needed to start a systemic therapy for metastatic castration-resistant prostate cancer as any line of treatment from the first line of treatment to the fourth line of treatment. So in this study, we would let people enter as initiators of pretty much any line of treatment. They need to have a good performance status from 0 to 2, because otherwise you're not candidate to receive cancer-specific treatment. They need to have bone metastases, no prior use of radium-223, and no prior participation in a trial for which unblinded information is not available. The treatment strategies in this target trial are the following. And whenever I talk about target trial emulation, I always like to make emphasis on the importance of properly specified treatment strategies. And this is one of the examples that I use often, because treatment in cancer is complex, because it's composed of several lines of treatment, and you need to reflect that on your treatment strategies. And this is what we try to do here. The experimental treatment strategy consisted of initiating radium-223, and then people are allowed to stop it. What are the conditions to stop it? Well, either you receive 6 cycles, because that's the maximum number of cycles that you can receive with radium-223, or you can stop it even earlier if you have toxicity, if the tumor progresses under this therapy, or if the patient has a worsening of their overall health status. What happens afterwards? We were also explicit about what they can do afterwards when they stop the treatment. We said that patients could continue other systemic drug for prostate cancer whenever clinically indicated, but never in combination with radium-223. In this treatment strategy, people could only receive radium-223 as monotherapy, not combined with other drug. For the comparator, the treatment strategy consisted of initiating any other standard of care. And for prostate cancer, these are docetaxel, cabazitaxel, enzalutamide, abiraterone, and some other drugs used in the country where we did this study. And patients are also allowed to stop the standard of care that they started and continue with other lines of treatment, with the exception of radium-223 as clinically indicated. Why is it important to be so explicit about what they can and cannot receive, and after baseline in the treatment strategies, because that will have implications on how we estimate the per protocol effect and on the interpretation of the actual effect that we obtain at the end. In our target trial, treatment assignment would happen at random, and patients would be aware of the strategy that they received. The outcomes are aligned with the objectives, are fractures, death, and prostate cancer-specific death. The follow-up starts at baseline and ends whenever we have an outcome, or 36 months after baseline. The causal contrast is the per protocol effect. Why is it a per protocol effect? Because this is a safety study. Because this is a safety study, we want to estimate the effect under full adherence to the treatment strategies. Because otherwise, if we implement an intention to treat analysis and adherence is very low, we might end up concluding that the drug is safe just because people don't take the drug. And we don't want that. We want to estimate the effect under full adherence. And in our target trial, to estimate the per protocol effect, we would censor patients when they deviate from the assigned treatment strategy, and then apply inverse probability weights to adjust for pre and post baseline prognostic factors associated with adherence. In practical terms, when do we censor patients here? Well, in the radium-223 group, in the experimental group, we censor them if they combine radium with another drug, because in the treatment strategy, we said that we are not allowing people to combine radium-223 with another drug. And in the comparator group, we censor patients if they initiate radium-223, because in the comparator, we don't allow people to receive radium-223. Okay, so we have our target trial. Now we need the data. In our case, we use a database from Sweden called PCBase. And this is a database that is composed by many pieces. The first component is the National Prostate Cancer Register of Sweden, which is a register where if you have prostate cancer, you are registered by law. It has a long history, exists since 1998, and it has very comprehensive information around the date of diagnosis, which on the one hand is great because you have a lot of information related to cancer characteristics, diagnostic workup, and primary treatment, but only about the date of diagnosis. In our trial, we want to estimate effects of a lot of treatments that happen way after one is diagnosed with prostate cancer. So we might need more information on top of this one. That's where the patient overview prostate cancer comes into play. This is another data set in Sweden created in 2014 to overcome the lack of follow-up data in the primary registration of the National Prostate Cancer Registry. It registers longitudinally treatments, laboratory values, clinical data, etc. And at the time when we analyzed data, which was 2021, this component of the PCBase data set contained data on approximately 12,000 patients from 33 healthcare providers. So it's a very rich data set. Also, important for our study, the contributing centers for the PPC covered pretty much all the sites that administer radium-223 in Sweden. So we had pretty much a complete capture of the administration of radium-223 in Sweden. PCBase also uses information from other registries in Sweden, the Cancer Register, Cause of Death Register, Prescribed Drug Register, etc., which complemented the overall data set. And as you can see, it was a very rich data set in Sweden. Using the Swedish PCBase, we extracted information on the presence of bone fractures using ICD-10 codes and the patient register, and information on the cause of death was available through the cause of death register. Because of the health system in Sweden, it was relatively safe to assume that if a If a patient has a fracture that requires medical attention, it would be there. And that because of the coverage of the health system, the follow-up is complete and comprehensive for pretty much everyone who stays in the country. So once we have the targeted health specification and our data set, now we can complete the data with the target health emulation column. For the eligibility criteria, we plan to use pretty much the same as in the target trial. We assume that everybody who started radium-223 had bone metastasis, and for the comparator, we included only people with recorded bone metastasis. This, you might feel a little uneasy here because we are applying different eligibility criteria to both different groups. And because of that, we had a sensitivity analysis where we only analyzed people with a recorded bone metastasis, and the results didn't change. The treatment strategies are the same. Regarding treatment assignment, patients are classified according to the strategy that they are compatible with a baseline. Outcomes follow-up are the same. The causal contrast is the observational analog of the protocol effect. And the statistical analysis is also the same with some additional adjustment for baseline covariates, because of course we don't have a randomization. And we also use sequential emulation, and I will explain that in a couple of slides. So we have the research question specified as the target trial. We have our data. We have a plan to emulate the target trial, and we were aware of 3 problems that we need to address via study design / data analysis. We'll see that we will find more problems. So the first problem was that we anticipated was that one of the eligibility criteria was initiating any systemic therapy for metastatic castration-resistant prostate cancer, going from the first line to the fourth line. Patients in PCBase in this database are observed for several lines of treatment, and actually this diagram on the right is a Sankey diagram of the actual patients in PCBase, how they migrate from one drug to another through several lines of treatment. So when should I include them? When they start the first line, the second, the third, the fourth. So we'll see how we handle that. The second problem is that we don't have randomization. And the third problem is that we are estimating the per protocol effect, and the artificial censoring that we impose to estimate that a causal contrast might be informative. So for the first problem, which is a problem of multiple eligibility, you can imagine how we handle that. We created line of treatment specific cohorts. Line of treatment was defined as the initiation of a new drug for prostate cancer. This could be docetaxel, radium-223, cabazitaxel, abiraterone, etc. And that dictated the initiation of a new cohort. So we created 4 cohorts. A cohort for first-line treatment, cohort for second-line treatment, third and fourth. What's important here is that at the beginning of each cohort, we extract the baseline characteristics, because those clinical characteristics will change over time and over lines of treatment. We assess eligibility at its baseline, and at that baseline is when we assign patients to one arm or the other. When we explained this in the protocol that was going to the EMA, we even added a toy example that we can see on the left, where we had 3 example patients with made up data and how they would contribute to the different cohorts. So the reviewers in the EMA could see a toy example of how those cohorts were constructed. We never got any pushback with this approach, so I think this worked very well. The second problem is the potential for baseline confounding. We dealt with that by adjusting for baseline variables. And one of the advantages that we had here is that this database was very rich on baseline confounders for prostate cancer. We had pretty much everything that an oncologist uses to decide what treatment to administer to the patient. The TNM stage, tumor grade, performance status, PSA level, hemoglobin, clinical conditions. So in this sense, the database was very rich. The third problem was, as I said, because we estimate the effect under full adherence, the artificial censoring that we impose might be informative, and we need to adjust for post-baseline characteristics. And in this sense, we were also lucky that we had time-varying information on important confounders like performance status, PSA, hemoglobin, osteoporosis, comorbidities, site of metastasis, prior treatment. So longitudinally, we were able to characterize patients very well. Okay, let's have a look. So we have a plan for the analysis, right? Let's have a look how the results came up. This is a very simplified flowchart of the selection of the patients into the study. We see on the left patients in the radium 223 group, on the right the comparator. Let's look at the left first. We excluded patients with missing values. The analysis was a complete case analysis. And then on the right, we have the label comparator, about 3,000 patients. Again, we selected complete case population. And I'm sure you have never seen this box in any flowchart ever before. This is shocking. How and why are we excluding people with immortal time? What are we talking about? Well, the fourth problem was a problem that we didn't expect, and was a problem of how the data was being generated. The data were generated in different ways for people in the experimental group, people receiving radium-223, than for people in the comparator group. In the sense that people who receive treatment other than radium-223 were only in the database based on one condition, which is that they were alive at the time when the center that were recording data on that patient was open. So patients need to be alive when that center was recording the database, recording data for these patients. So that is represented in this diagram on the right. So in this diagram, we have three patients, ID1, ID2, and ID3. For ID1, this is a patient that received several lines of treatment, first line of treatment, second line of treatment, and third line of treatment. But this is the date when the center that entered the data about this patient joined the database. That center joined the database when that patient was receiving the third line of treatment. But this center also recorded the information of this patient retrospectively. Because I didn't know that, I created a first line of treatment for this patient, a second line of treatment, and a third line of treatment. And by doing that, this is immortal time. I was granting this patient immortal time from the second line and the first line of treatment. It's immortal time because he's only here if he was alive when the center joined the database. If we contrast this with a second example, ID2, another patient like the first one who starts first line treatment, but then he dies, this patient is not in the database because he's not alive when the center joined the database. If we look at a third patient who received Xofigo, patients receiving Xofigo are in the database regardless of whether they were alive when the center entered the database or not. So if this patient dies early, that patient is on the database. So you can see that we are getting rid of people who in the comparator group die early, and we are granting immortal time to some patients. This is a bias. This is a bias that fortunately we learned about, and that also had an easy solution. It means that you only include data after a date when a center joined the database. But I think this highlights the importance of working closely with people who are true experts on the database or how the data were generated. So yeah, once we handle that, and you may be thinking, how did you figure that out? Well, we figured this out because we were obtaining completely implausible effects on survival. In prostate cancer, this is a very common tumor, and you have very accurate expectations of survival by line of treatment, by age, by stage. That's very well characterized. When you analyze a database that has survival data completely not aligned with what we know about the tumor, something's going on. And that's what we found, and that's why we kept digging in and talking with experts on the database until we realized this was happening. Okay, let's have a look at the results. I'm not going to go over all the numbers, but I want to highlight some results here. Here we have the results by line of treatment, first line, second line, third and fourth line. And the first thing that we can see is that the main study outcome, which is bone fractures, we don't have that many events. First line, we have 15, 31. Second line, 25 and 8. And then third line and fourth line is where we run into problems, because in the comparator group, we only have one outcome. In the fourth line, we have 0 outcomes. With this, you cannot really model any effects in these late lines of treatment, which was a limitation. These are the baseline characteristics. I'm not going to go over all the details. And these are the main results. So for the first line cohort, the risk of fractures, we estimated the effects using cumulative incidence curves. And we did this on purpose because we wanted to first be able to estimate absolute risks, and you get that with a time to event curve, and then you can use those absolute risks to estimate risk differences and to estimate risk ratios, as opposed to giving just only one hazard ratio. And also, cumulative incidence curves or survival curves can serve as a tool to diagnose whether something is weird with your data. If you see very early separations, something might be happening. So that's why we decided to go with survival curves. For the first line of treatment, we estimated that was an excess risk of about 6.5% for patients receiving radium-223, although you can see that the confidence interval is wide. For the second line treatment, the excess risk was 7.6%, also with wide confidence intervals. And for the third, fourth line of treatment, we didn't model the effect. We just provide a description of the outcomes, because we only have one outcome combining both cohorts. So you don't get meaningful results with modeling this. We also ran a pool analysis. We combined together the 4 line of treatment. This was pre-specified in the protocol. In the protocol, we said that we were going to estimate the heterogeneity of results with an I-square, and if it was less than 50%, I think we would combine the results, and that was the case. So we combined the results, and when we combined the 4th line cohorts, the excess risk of fracture was 8.7%, and you have here the confidence interval, and also the hazard ratio of 1.61. Okay, that was for the risk of fractures. So there seemed to be an increased risk, a moderate increased risk, but the confidence intervals were not very precise. In terms of overall survival, in the first line of treatment, we obtained these weird results where we had an increased risk of death, an increased risk of 13%, risk difference of death at 3 years of follow-up. In the second line of treatment, there was the signal was not there. The risk difference was minus 7.6%, but the confidence interval, you see how wide it is, so it's not very informative. And for third and fourth line of treatment, look at the confidence intervals; they are all over the place. So not very informative results. But it was concerning that the first line there was a signal towards an increased risk of death. With these results, we went to the clinicians and talked to them, and they say, of course, this first line comparison doesn't make a lot of sense, because here we are comparing radium-223 as monotherapy versus another standard of care. As I mentioned at the beginning, radium-223 is not a systemic treatment for prostate cancer. It doesn't treat the whole tumor, let's say. It only targets the bones. So if you have a patient in a first line of treatment, you want to start with a drug that treats the whole tumor, that treats the tumor systemically. If you give only radium-223 as a first line, that means that that patient has a reason for not receiving full treatment for prostate cancer. So these are probably very frail patients. And we have a very comprehensive characterization of patients at baseline. We have performance status and other variables. But frailty is different from performance status. You can have a relatively good performance status, but the doctor might perceive that you are frail for other reasons, and that is not in the database. So our concern here was a measure confounding. We were not comparing apples to apples, and something what's going on like a measure frailty. So to address that, we resorted to a negative control outcome. We chose to use a composite cardiovascular event because radium-223 has no effect on cardiovascular events, no effect at all. And we argued that, well, the measure confounding, the structure of the measure confounding could be similar for death than for cardiovascular events. When we ran this analysis, we, again, all the analysis had very, very wide confidence intervals, but the overall trend was very similar. At first line of treatment, there was an increased risk of cardiovascular events in patients receiving radium-223, and that's clearly not because of the drug, has to be because of another reason that could be the reason also biasing our results for overall survival. So yeah, that's how we designed the study, though that's how we implemented it, the problems that we encountered and how we addressed those. And this is how we discussed the results to the EMA. First, we compare our estimated risk of bone fractures among people who received radium-223 in our observational cohort. And we saw that we had about 4% risk in the first line cohort, 5.8 and 5.3 in the second line cohort and fourth line cohort, and compare these to similar populations. In the Alcinca trial, which was the pivotal trial, the risk of bone fractures of patients receiving radium-223 was about 6-7%, so we were on the ballpark. And in the ERA-223 trial, which was the trial that used radium-223 earlier as a first-line therapy, the risk was between 4-5%. So again, we were in the same, more or less estimating the same magnitude of the risk of fractures associated with radium-223. When we estimate effects of radium-223, we are comparing it with other comparator, and the increased risk that we are reporting, we thought was driven a lot by the low number of fractures in the comparator. So in the second line cohort, there was only 8 fractures. That was a 6-month risk of 3.9%. In the third line cohort, there was only 1 fracture. That means a risk of bone fractures of less than 1%. And as I said before, prostate cancer is a pretty common cancer. It's very well characterized, and we know that there is a risk of bone fracture in these patients. For example, a study using CR Medicare estimated that the 6 months risk of fracture was about 7%. So the bone fracture risk that we were obtaining in the comparator group was not aligned with what is known for this disease, and that was driving our effect estimates. Why was that happening? I think one of the reasons is that our follow-up was relatively short in the comparator group, and part of the reason why we had that short follow-up was because we had to exclude that time, that immortal time. So we didn't have a lot of follow-up, and that you might be thinking, well, it doesn't matter. You have less follow-up, you also have less events, and your risk is events over follow-up. But when you have less follow-up, the opportunity for random variation opens is greater. So I think that those are factors that could be playing a role here. That was about fractures. Related to overall survival, what we discussed, we had this weird result on first-line treatment, and the interpretation here was that patients receiving monotherapy with radium-223 are a specific case, are patients who are not candidate to receive the standard first line of treatment. And I mentioned this at the beginning. The standard first line of treatment is not very toxic in prostate cancer. Because the standard first-line treatment are second-generation anti-androgens, are not chemotherapy. So they have toxicity, but they are not as toxic as docetaxel. So if somebody is not even candidate for that those are special patients. And we try to reflect this with this negative control outcome analysis, where we reproduce the associations that we're obtaining for survival. So we concluded that using real-world data, we were estimating a risk of fractures related to radium-223 aligned with the risk observed in randomized clinical trial, that our effect estimates might be influenced by the low rate of bone fractures in the comparator, and that our estimates on overall survival had to be taken with a grain of salt because of the potential of residual confounding. So key takeaways from this case study plus the two documents that we review at the beginning. Causal inference is now recognized as a data science task by regulators. I think now you can be explicit on your protocol that you are emulating a target trial, and you can provide the target trial table. If you review the protocol that I wrote, that it's on the public domain, there is no mention to target trial emulation. There is no table about target trial emulation, because we wrote that in 2019 before these documents were out. So you can see that those protocols contain all the elements of the target trial. They are explicitly describing the treatment strategies. They have the causal contrast. They have all the components, but they don't mention target trial emulation because at that time I was not sure the regulator was ready to accommodate that framework. But after these two documents that we discussed at the beginning, I think it's safe to frame your post-authorization safety study as completely as a target trial emulation. We've seen how we implemented this in this case study, and this study met the regulatory requirements. And in cancer, I think it's in post-authorization safety studies of cancer. I think the target trial emulation framework is especially useful because treatment is complex and changes over time. Patients receive several lines of treatment. The clinical characteristics change. So specifying properly the treatment strategies, I think, is very important. And also being clear about your causal contrast, the effect on the full adherence, and how you implement that is also important and can be complex in cancer. And using the target trial emulation framework makes your life easier. And that's all I had. Now I'm happy to take questions. And if you don't have questions now, but you have questions later, this is my email. Feel free to send me your questions.